So you might be wondering what happens to these mesenchymal cells that are created by the primitive streak on the dorsal side of the embryo. And they're going to migrate down and create a new layer in the embryo, but that's not all they do. So these mesenchymal cells are pluripotent. They can become any of these cell lines. Some of them are going to indeed make the middle layer of the embryo called the mesoderm, and we're going to even more specifically call it intraembryonic mesoderm because there is mesoderm that's extraembryonic, and we'll touch on that again later. Some of these mesenchymal cells that proliferate from the primitive streak are actually going to replace the hypoblastic cells and become the embryonic endoderm. And the remaining epiblastic cells that were on the dorsal aspect of the embryo are going to become ectoderm. So these mesenchymal cells don't only become mesoderm, they also replace the hypoblastic cells, and some of them become the ectoderm of the embryo. All right, so at the same time that this primitive streak is developing and adding growth to the caudal end of the embryo, we also have the development of another anatomic structure that's called the notochordal process. So some of these mesenchymal cells that migrate to the midline of the embryo isn't going to develop in mesoderm there, it's going to develop into a special structure called the notochordal process. And so this is a dorsal view, and you can see here that the primitive node pit is going to stop at this location, and all of the middle layer of the embryo that resulted from migration of mesenchymal cells in this region is not going to develop into mesoderm. It's going to develop into this structure called the notochordal process, and this is what it looks like here. This is a mid-sagittal cut through the embryo, so it's directly in the middle. And the primitive pit and primitive node is actually going to expand downwards into this developing notochordal process, as you'll see in 1 second. And so it is going to actually span this notochordal process between the precaudal plate, or the area where the precaudal plate was cranially, and I'll show you a picture of that here. So this is where the precaudal plate, that elongated and so longer columnar cells is located at, and we're actually not going to have any migrating mesenchymal cells in this area. So this precaudal plate will remain 2 layers in the embryo, and it is going to be the future site of the oropharyngeal membrane, which you'll learn about when you learn about the gut development and development of the oral cavity. So this will remain 2 layers in the embryo. And then on the same token, on the other side of the embryo, on the caudal aspect or the tail end, we also have an area of the embryo that only has 2 layers. And that's called the cloacal membrane, and that's going to be the future site of the anus. So we have the area that's going to be the beginning of the gut tube, and we have the area that's going to be the end of the gut tube, both remaining to be only 2 layers in this embryonic disc. So the notochordal process is in the midline, and it's going to span in the midline all the way from that precaudal plate, where there's only 2 layers, to the cloacal membrane. And so this pit that was the primitive, where the primitive node and primitive pit located, the depression where the primitive streak had started, is going to actually depress downwards and into the notochordal process to make what's called a notochordal canal. So if I click here, you can see it expand a little bit more as the notochord process grows. So does this notochordal canal, which is actually the expansion of the primitive pit. And I click it again, and you can see further growth and further expansion of the notochordal process and also the notochordal canal. All right, so interestingly enough, it's not going to remain in this configuration, but rather the floor of this notochordal process is going to fuse with the endoderm. So the yellow layer here is going to fuse with the floor of the notochordal process, and parts of this floor is going to degenerate and have open communication between the umbilical vesicle here and the amniotic cavity. And if you think we have growth and a lot of changes in pressure, we might want to have communication. So this actually further expands and the floor will completely degenerate away so that we will end up with complete communication between the two. So after this happens, we're going to have a small little area of open communication, but it actually lingers a little bit longer as the rest of this notochordal process is going to reconstitute itself and make a continuous layer. We still have a little bit of equalizing and communication of pressure between these two cavities, and that's thought to be the purpose of having this communication. Of course, everything has a special anatomic name, and this little communication or hole between the amniotic cavity and the umbilical vesicle, you'll see it in the textbook, it's called the Neurenteric canal. So don't get confused, for that's the fancy name for this open communication between the two. And so now I have switched to a transverse or axial view through the embryonic disc. So this image before, this was a longitudinal cut, so you could see it was just mid-sagittal. So this would be the cranial and this is the caudal region, this is the cranial region, and then we had this notochordal process growing cranially and the primitive pit expanding cranially. Now I'm going to switch to a transverse view, so I've cut the embryo in transversely or an axial view, and so now you're looking at that area where that notochordal process had developed and the floor degenerated. You can't see the open communication because we're not at the neuroenteric canal area, and here is the ectoderm, and we can see the mesoderm in this region here, and this is the cells that belong to the notochord. And so what happens is that this plate of cells that's left is going to actually involute, and so I'm going to click and you can see how the edges come into close approximation with each other, and they're going to fuse at the midline, and it's going to create a complete restoration of the endodermic layer here, and it's going to create a circular structure at the end that we call the notochord, now that it has, those ends have come together and fuse. And so this structure is at the midline of the embryo. Now you can see that the rest of this on the sides is just going to be mesoderm, but this is not mesoderm. This is special cells that do not have mesodermic fates, but rather they do what the notochord is supposed to do, which we will talk about in 1 second. And so, in a summary, by the middle of the 3rd week, after we've had migration of all the mesenchymal cells, there's going to be mesoderm everywhere between ectoderm and endoderm now that these mesenchymal cells have migrated, except for in 3 places. At the oropharyngeal membrane cranially, that's going to become the future site of the opening of the foregut. At the cloacal membrane caudally, which is the future site of the anus. And in the median plane, in the middle of the embryo, where that notochordal process developed. So every other place is going to have mesoderm. And so now we can answer a couple questions and we can continue on with our studies.